Hi team, welcome back to the lab. And some of you needed to know a little bit more information about cytokines, how they work, and to know about all the different kinds that we need to know for this class. So I want you guys to remember, always in general, whenever we talk about any kind of signaling, we're going to talk about, up here in this corner, our general diagram, which is we have a signal, and we're only talking about protein signals in this class. That protein signal is going to dock at a receptor, and then that binding together of signal to receptor means that the signal is brought inside the cell. The signal is transported down into the nucleus, where that's going to interact with our DNA to turn on or off production of certain genes into proteins, and then finally that's going to elicit some kind of response. So here in the corner I've translated this into a very general picture of how immune signaling happens, and the particular kind of signal that we're going to be talking about are called cytokines, and that's just the other word for immune signals. And these cytokines, just like in our general model, are proteins. And so, in general, the cytokine is going to bind to its cytokine receptor. The signal is going to be brought inside the cell, into the nucleus. Certain genes are going to be turned on or off to be made into proteins. And in the end, we're going to have some kind of immune response. And when we talk in general about these cytokines, we have to realize that there are actually hundreds of different kinds of cytokines that all when bind to the receptors, do slightly different things to the cell that it binds to. For this class, we're only going to learn about 5 different cytokines, so that's something to keep in mind. So here's one example over here. This is a very general picture of what could happen with a cytokine signal. So one class of cytokines are called interleukins, and right here I've drawn interleukin 2, or we can say IL-2. And in general, what's going to happen with this cytokine is that it's going to bind to the IL-2 receptor. And just as in our general model, the same thing happens. The signal is brought inside the cells, the nucleus, genes are turned on or off, and this elicits some kind of an immune response. In the case of IL-2, this is a pro-T cell cytokine or signal, so it's going to make more T cells, so the cells are going to divide. They're going to also make and release more cytokines. If this were in particular a killer T cell or CTL, it would activate to go search and destroy for some infected cell. So yeah, different cells, different functions, and they can be turned on by this IL-2 signal. So on the next slide, we're going to get into a few examples of these cytokines and how they work on different cells. Okay, we are back and we are getting into our examples of different cytokines. In the PowerPoint core concepts slides that I gave out last week, there is a table with the list of cytokines that you ought to know for this class, because they're the most talked about when we get into talk about vaccine science. So those are the ones to focus on. You don't need to memorize the hundreds of other cytokines that are out there. And the big thing to remember is that cytokines are protein signals. So if you can just remember our general model of signaling, that model applies for all of our cytokines. It's just that the response is going to be different depending on which cytokine we're looking at. So here's a specific example. So here's a killer T cell, or a cytotoxic lymphocyte, and I've drawn on its surface an IL-2 receptor. So in the cytokine or signal is our IL-2. So IL-2 is going to bind to an IL-2 receptor on a killer T cell, and the signal is going to be brought inside the cell to the nucleus, and then there are going to be specific immune response actions that happen after that. So IL-2 stimulates the CTL to go find the infected cells and kill them, but also wants to make more of itself, so it gets activated to divide and proliferate. It also could tell it to migrate to the site of infection, And it also wants to release more cytokines so that it can enhance the immune response once it gets activated. Our second cytokine that we're going to talk about, this is the cytokine that I actually work on here in the lab, and it's called IL-10. And unlike IL-2, which acts to activate CTLs, IL-10 does the opposite to T cells. It actually shuts down the T cells. It suppresses them. So IL-10, the cytokine signaling molecule, is going to bind to its receptor, which we're going to call IL-10 receptor. The signal is brought inside the cell. Genes are turned on and off. And in case of our helper T cell type 1 here, instead of the IL-10 activating the cell, it's actually going to shut it down. So the immune response actions here are to stop cell division, to initiate apoptosis, which is the regulated programmed cell death, and also it will release more cytokines to suppress itself. And definitely stop the production of pro-T cell molecules like the IL-2. So if you just can remember that IL-10 does the opposite of IL-2. IL-10 suppresses the immune response. IL-2 promotes the T cell response. Okay, so on to our third example down here in the corner. We have our B cell here, and on the surface of the B cell is an IL-4 receptor. So IL-4 is another cytokine that works to promote the antibody response. It works to promote B cells. So the IL-4 signal binds to our IL-4 receptor. The signal is brought inside the cell, into the nucleus. Genes are turned on and off, and we have a specific immune response that happens. So in this case, IL-4 elicits B cell proliferation, so that's cell division, making more B cells. It's also a go signal for that B cell to make more antibodies. It's like, oh, we've got the right antigen-antibody combination here, let's make more of that. It also promotes differentiation or specialization into our plasma cells, which, if you remember, are the antibody factories. And then in our last example over here, I just wanted to point out that these receptors are not necessarily on just one kind of cell. So I've shown here that the IL-2 receptor is not only on the killer T cells, like in our first example, but it also can be found on the Th1 cells. So it's very similar to the killer T cells. The IL-2 will bind to its IL-2 receptor on the helper T cell. Transduction will happen, the signal will be brought into the nucleus, genes are turned on and off, and a specific immune response happens. Now, because this receptor is found on the Th1 and not the CTL, a slightly different response happens. So we do get a promotion of cell division and release of cytokines, but since this isn't a killer T cell, it's not activated to go kill infected cell. But since it's a helper T cell, it's activated to do its job, which is to make more cytokines, like it could make more IL-2, and then to make more of itself as well. So we're going to talk about more of this in class, and there's going to be one group presentation on this next week. So we'll get into a little more applications of this biology. The bigger picture is to remember is that there are hundreds of different kinds of cytokines. They each do a slightly different job. Some can promote the T cells, like killer T cells and Th1s, but some are actually going to promote the B cell response too and making more antibodies. And another class, like the IL-10 cytokine, will work to shut down the T cell response in Th1s and CTLs. So it can get a bit confusing. Please email me with any questions you may have.